In our lab at Penn State University, we study human herpes viruses 1 and 2, which are better known as herpes simplex virus, or HSV. HSV is best known for causing oral and genital lesions that recur again and again. Oral HSV infection is very common, with about 70% of adults in the USA showing evidence in their immune system of past or present infection by HSV-1. Genital herpes virus infection is less common, but it is on the rise. At present, about 1 in 5 adults in the USA have been exposed to genital HSV. The Centers for Disease Control estimate that 750,000 new genital HSV infections occur every year, with about half of these afflicting young people under age 25. Since HSV infection lasts a lifetime, the number of HSV-infected people will only increase unless we find a way to block viral spread or to vaccinate people. HSV is a large DNA virus with a protein layer called the tegument, which is surrounded by a fatty lipid envelope. That envelope makes the virus infectious, but it's also susceptible to drying out. Hence, you can only get HSV through close, wet contact, such as kissing and sex. What makes HSV a troublesome virus is that it doesn't stop at causing skin or genital lesions. In addition to replicating at the skin where you can see and feel it, the virus also gets picked up by your nerve endings. Your nervous system includes your brain, spinal cord, and the peripheral nerves that connect to all parts of your body. Inside your nerves and neurons, HSV establishes a special latent form of infection where it goes silent for a while. That while might be as short as a week or as long as a decade or a lifetime. The virus can reactivate at any time, usually when you're under stress, and exit via the same nerves that it first entered to reinfect the skin and reach a new host. The reason that infection lasts a lifetime is because HSV's latent infection in nerve cells, or neurons, can never be cleared by your immune system. Physicians can prescribe a drug called acyclovir to treat the lesions you see and feel on your skin, but that drug only works on actively replicating virus, not the latent stage in neurons. Acyclovir and its derivatives essentially function as a band-aid treatment for infection. They shorten the lesion duration and help you heal faster, but they don't treat the underlying source of infection. For that, we need to develop therapeutics that work in neurons, by blocking reactivation or boosting the immune response to control the infection. Our lab is working to build resources to do just that. Here in our lab at Penn State, we are furthering the field of HSV research in 2 ways. First, by pushing forward our ability to work with neurons to the point where we can screen for drugs that block infection in neurons. And second, by using new genome sequencing techniques to understand how different HSV is around the world and from person to person. First, let's talk about our research with neurons. As we mentioned earlier, the main drug used against HSV is acyclovir and its derivatives. We don't have an antiviral drug that interferes with latency or reactivation in neurons. This is largely because human neurons are not widely available and are difficult to grow in the lab. Our lab is working to develop easily reproducible protocols to work with human neurons and to study HSV infection in these neurons as a prelude to using them for future drug screening efforts. We will be studying the normal characteristics of how these neurons grow in the lab, and then looking at how the neurons are changed by HSV infection. Then we can add various drug candidates into the system of HSV infection in neurons and ask which drugs can interfere with the process of infection and at what point the viral infection gets blocked by a drug. Importantly, we can also screen for side effects on these neurons and eliminate early on any candidates that produce harmful outcomes for neuronal health. In addition to our work with neurons, we also use high-throughput genome sequencing to study differences in HSV strains. Just like people, every virus is different. Subtle variations in the DNA genome of the virus can lead to differences in the outward characteristics of each virus, just like people differ from one another. Our lab has pioneered the use of new high-throughput genome sequencing techniques to reveal diversity in HSV strains. On a global scale, this has shown us how HSV strains vary from country to country. For instance, this phylogenetic tree of multiple HSV strains demonstrates how the DNA sequences of viruses from the same country look more similar to one another than to HSV strains from other countries. Viral genome sequencing also works on a personal scale, for instance by allowing us to see how one person can shed viruses that vary subtly from one another. Here we can see a fragment of DNA sequence from 2 separate HSV strains that had infected the same person. These 2 strains differ in key residues that may affect the function of viral proteins. Deep sequencing of viral genomes will reveal more insights about how HSV strains vary from person to person and within one person. If we can make viral genome sequencing rapid and accessible enough, then we can envision one day having personalized medical treatments that are specific to the particular virus strain that a given patient is carrying. This idea could apply to HSV or to any number of infectious disease agents. For now, we hope to use our new knowledge of viral diversity to improve vaccine and drug efficacy, so that the next generation of therapeutics are designed to combat not just one, but rather all strains and types of HSV. Thank you for listening, and I hope you enjoyed learning about HSV and the research in our lab at Penn State.